My name is Caroline Dipzinski. I am a senior biology student here at Presbyterian College, and I will be presenting my honors research on the induction of chemotherapy resistance with drugs used in the treatment of mental illness. So I've actually been working on this project since June of 2021. I first joined it through SE Embry and just kind of stuck with it since then. It was an ongoing project when I joined, so the very first few steps of the research I actually was not there for, but this presentation will kind of show you what I did contribute to the research. So I'll first go through some background information, our study aim and our hypothesis, our methods, the results obtained, a bit of discussion on those results, and then future direction and acknowledgements. So this project basically came about after several studies showed that there was a higher case fatality rate from cancer in psychiatric patients. And there's actually quite a few factors that could kind of explain that finding. People with severe mental illness were found to be less likely to receive routine cancer screening, in turn leading to a later stage at diagnosis and a more advanced disease at diagnosis, making it harder to treat. It was also found that sometimes the symptoms of mental illness and the medications used to treat that mental illness could mask the cancer symptoms, again leading to a delay in diagnosis, again that later stage and more advanced disease at the time of diagnosis. And even after diagnosis, patients who suffer from both mental illness and cancer were found to have a more difficult time navigating their own treatment due to their depression, anxiety, whatever mental illness they may be suffering from. However, this research kind of focuses in on multiple drug resistance as an explanation for that finding. Multiple drug resistance is defined as the failure to respond to multiple drugs with different mechanisms of action. And as you can see in Figure 1, there are several factors that can cause multiple drug resistance, things such as decreased influx of drug into the cell, the activation of detoxifying systems, blocked apoptosis, the activation of DNA repair, and what this research focuses on, the increased efflux of drugs from the cell through ATP-dependent efflux pumps. P-glycoprotein is one of those ATP-dependent efflux pumps involved in multiple drug resistance. It is an ATP binding cassette transporter, or an ABC transporter. It's actually one of 48 ABC transporters. However, this one was chosen because it was one of the very first ABC transporters identified to be associated with multiple drug resistance. P-glycoprotein is the product of the ABCB1 or the MDR1 gene. Those names are used interchangeably. It is 1,280 amino acids long, and as you can see in Figure 2, which is the two-dimensional structure of the human multiple MDR1 P-glycoprotein, it has 12 transmembrane segments, which are thought to form together to form a 3D barrel-like structure in the membrane, allowing those drugs to be pumped out. P-glycoprotein is primarily found in the blood-brain barrier and the intestine. It has protective properties in both areas by transporting drugs from either the brain or intestinal side to the blood side, preventing the accumulation of compounds and drugs in those areas. So again, it actively extrudes a variety of drugs. It has a broad substrate specificity, making it resistant to the cytotoxic activity of many drugs. So as it relates to this research, many drugs used in the management of mental illness are actually substrates for P-glycoprotein. Likewise, several chemotherapeutic agents, such as vinca alkaloids and taxanes, are also substrates for P-glycoprotein. So it's thought that multiple drug resistance as it relates to mental health and cancer is due to the long-term use of mental illness drugs that are substrates for P-glycoprotein, essentially building up that resistance to the chemotherapies when also diagnosed with cancer. With this information, this study aims to determine if the long-term treatment with drugs used to treat mental illness can contribute to the development of drug resistance in human cancer cells in vitro. We hypothesize that the long-term treatment with drugs used to treat mental illness can contribute to the development of drug resistance in mental health patients by upregulating the ABCB1 gene, resulting in the overexpression of P-glycoprotein, leading to the poor prognosis of patients who suffer from both mental illness and cancer. To test this hypothesis, CACO2 cells were cultured in vitro and administered a high and low dose of various drugs. CACO2 cells are a human colorectal adenocarcinoma cell line. They are a good cell line to study drug transport because they form tight junctions with each other, kind of minimizing the factors involved in drug transport. So cells were cultured under sterile conditions, fed minimal essential media once a week, supplemented with FBS and penicillin streptomycin. Cells were split once a week as well, and upon every split and feed, cells were administered a low and high concentration of the drugs listed in Table 1. These drugs were chosen from the 2017 Top 300 Drug List. Drugs were then filtered by the use for the management of mental health, and from there sorted based on their affinity for P-glycoprotein. So all the drugs selected are substrates for P-glycoprotein, including vinblastine, our control, and dimethyl sulfoxide, another control. Atomoxetine is a selective norepinephrine reuptake inhibitor, or an SNRI, used in the treatment of attention deficit hyperactivity disorder. Levetiracetam is an anticonvulsant agent used to treat patients with epilepsy. Olanzapine is an antipsychotic used to treat patients with schizophrenia and bipolar I disorder. Vilazodone is an antidepressant used to treat major bipolar disorder, and vinblastine is a vinca alkaloid, anti-cancer drug, and DMSO, dimethyl sulfoxide, is also used to treat cancer. So again, these drugs were administered over a long period of time to determine if their long-term use would in fact induce the resistance to chemotherapy through overexpression of ABCB1 and in turn P-glycoprotein. To determine the changes in expression in ABCB1, we set up quantitative PCR for each of our cell treatment groups. We first started by extracting RNA from each treatment group and using that RNA to synthesize cDNA using a cDNA synthesis kit. Reactions were then set up using primers for the ABCB1 gene to select for that gene. The cDNA was then added to a 96-well plate organized by drug treatment and analyzed in a real-time PCR thermal cycler. From there, fold changes for each sample was determined using Microsoft Excel and graphed in Figure 3. So as you can see, both high and low doses of vinblastine demonstrated a high expression in ABCB1. Similarly, both high and low doses of vilazodone showed an increase in ABCB1 expression. The low doses of both atomoxetine and levetiracetam showed an increase in ABCB1 expression. However, as you can see in the graph, both low and high doses of olanzapine and high doses of atomoxetine and levetiracetam did not show higher expression rates of ABCB1. Proliferation assays were then set up for each cell treatment group to determine the proliferation rates of cells exposed to chemotherapy agents that are known substrates for P-glycoprotein. We used vinblastine, which is a vinca alkaloid chemotherapeutic agent, and docetaxel, which is a taxane chemotherapeutic agent, in our proliferation assays, both of which are substrates for P-glycoprotein. So essentially what was done was cells were plated in 96 well plates, and a 4-fold serial dilution of either vinblastine or docetaxel were added to each well of the plate. So plates were organized by column based off cell treatment group, and then by row based off concentration of vinblastine or docetaxel. So at the very top of the plate, that first row would have the highest concentration, and then as you go down the plate, the concentration gets lower and lower. Cells were then allowed to incubate for 48 hours in the presence of either vinblastine or docetaxel, and then WST reagent was added to each well of the plate to determine the number of viable cells. The way WST works is it detects the number of viable cells by detecting their metabolic activity. So as the cells are metabolizing that WST reagent, there is a color change, and that color change can be quantified using a spectrophotometer. Specifically, we use the cell imaging multimode reader, so you just put the plate in and it's read by a machine, and those results can be exported to Excel and graphed in a graph that looks like figure 4. So this is pretty much organized by increasing chemotherapy drug concentration across this x-axis and percent growth across this y-axis. We put both vinblastine proliferation assays and docetaxel proliferation assays on the same graph for each cell treatment group. So I kind of organized this by drug treatment groups, so this one will show vinblastine, and then the next atomoxetine, and so on. So I'll just go through the drug treatment groups and their associated proliferation assay results. Figure 4 shows the proliferation assay of cells treated with the low and high dose of vinblastine. As you can see, they demonstrated resistance to vinblastine, but were more sensitive to docetaxel. So the low dose of vinblastine, seen by this blue line, and the high dose of vinblastine, seen by this gray line, both showed, again, resistance to vinblastine in their proliferation assay, but these yellow and orange lines showed more sensitivity to docetaxel. So again, figure 5 shows the proliferation assay of cells treated with both low and high dose vilazodone, again with vinblastine and docetaxel proliferation assays graphed on this same graph. This pretty much shows that there was the most resistance to vinblastine in the groups treated with lower doses of vilazodone, seen by this blue line. Even at higher concentrations, they showed resistance to that vinblastine chemotherapeutic agent. The high dose of vilazodone showed some resistance. Again, at this high concentration, there wasn't much drop-off. And then the low and high dose were more sensitive to docetaxel. The proliferation assay of cells treated with either the low or high dose of levetiracetam demonstrated that there was resistance to vinblastine at the low dose only, seen by this blue line right here. The high dose of levetiracetam in the presence of vinblastine, this orange line, showed a lot of sensitivity, and both low and high doses of levetiracetam in the presence of docetaxel also showed a lot of sensitivity. Figure 7 shows the proliferation assay of cells treated with either the low or high dose of atomoxetine. This shows that there was resistance to vinblastine at the low and high dose of atomoxetine, seen by the red and orange lines right here in the middle, even at higher concentrations of chemotherapy drug the proliferation rate stayed fairly consistent. Interestingly, there was also resistance to docetaxel at the high dose seen by this yellow line. Again, those proliferation rates are fairly consistent, even increase a little bit through this area. However, the low dose of atomoxetine was much more sensitive to docetaxel, as you can see in this large drop-off, as concentration of chemotherapy drug goes up. These findings are yet to be explained by this research. Our quantitative PCR data showed that there was an increased ABCB1 expression for several drug treatments, specifically the low and high doses of vinblastine, the low and high doses of Vilazodone, and the low doses of both atomoxetine and levetiracetam. These results were actually consistent with Western blot data. However, that's one of the things that was performed before I joined the project, so that data is not shown in this presentation. Some explanations as to why there was higher expression in lower doses compared to higher doses could be that some drugs are actually inhibitors of P-glycoprotein at higher doses, but inducers for P-glycoprotein at lower doses. And that is actually consistent with other drugs that have been used in the same model. Our proliferation assay showed that their varying concentrations of drugs demonstrated resistance to vinblastine and docetaxel. However, more data is needed for further data analysis. We actually had a difficult time finding a protocol for the proliferation assay that provided reliable results. We initially were using Presto Blue, but found that the results we were obtaining were just very unreliable, very all over the place. So we tried out the WST a few times and found that it was much more consistent. So we worked on perfecting a protocol, because even the way that we plated the cells and administered the chemotherapeutic agents, vinblastine and docetaxel, we found varying results in the different ways that we did that. So we only had a few replicates for the proliferation assay, again, because we just kind of perfected that protocol for the proliferation assays using WST1, and again, just the way that the whole experiment was set up, the plating, the administration of the drugs. Future directions for this project would again to have more proliferation assays run to get sufficient replicates for statistical analysis. Like I mentioned previously, we have just kind of figured out a way to get good reliable results from those proliferation assays, so definitely more replicates. We would also like to get RNA-seq data to determine additional changes and expression induced by drug treatments in vitro. I would now like to acknowledge SC Embry for funding and support of this project, Dr. Amy Messersmith from the PC School of Pharmacy, who is actually head of this project. She has advised me throughout my entirety of working on this research. I would also like to acknowledge Dr. Stuart Gordon for helping me find this research opportunity. Thank you.